Bismillahirrahmanirrahim. Today we are going to learn about the mechanism of contraction of skeletal muscles. Almost all the muscles inside the body are having somewhat same mechanism of contraction. To know about the mechanism, we are going to the first slide. In this slide, we can see that actin and myosin filament are overlapping each other. This is the actin filament, while this blue color is the myosin filament. The actin filament is made up of tropomyosin, the thread-like structure, the tropomyosin, the bead-like structure, the actin, are also called F-actin, the bead-like structure, the black in color. This is the actin, and the red color troponin, which is having three domains. The troponin, which is having a red color and three domains, that is represented by red color. This troponin is further having three domains: the troponin T, the troponin C, and the troponin I. The troponin T domain is responsible for binding with the tropomyosin. The troponin I is responsible for binding with the actin filament, with the actin F, or F-actin, and tropomyosin and troponin C is responsible for binding with the calcium ions, which is liberated by the cell. So now coming towards the myosin, and the myosin filament is made up of two heavy chains and two light chains. The two heavy chains are twisted around each other, these two heavy chain, and making the head portion over here that is represented by blue color. This is the heavy chain, and the light chains are over here in the near the head and in the arm. This is the arm of the myosin, and this is the head of the myosin, which is made up of two head, and the neck area and the neck area and the arm area is having two light chain, the light chain in green color and the light chain in red color. Two types of hinges are there. The hinge one is over here, uh, beside the neck, below the first light chain protein, the first light, below the first light, in between the first light chain and the second light chain protein, there is hang one. With this hang one help in the attachment of this head with the actin, while the hang two is over here, that is in between the arm and the main stem. So this hang help the myosin to slide against the actin filament during the power stroke. The head of the myosin filament also contains two binding sites, the binding site for the ATP and the binding site for the actin. The actin binding site is responsible for binding with the actin filament, and the ATP binding site is responsible for binding with the ATP, and this also contains the ATPase enzyme, which is also responsible for the breakdown of ATP that is bind with this site. So coming toward the detail. So first of all, what happened? The myocyte that is connected with the neuronal terminal get the action potential from the neuromuscular junction, and this action potential is transferred through the sarcolemma of this myocyte. In this diagram, we can see that this is the action potential, and this action potential is going to be transferred throughout the sarcolemma. But when this action potential reaches this T tubule, it goes inside the T tubule, and all the way it's spread throughout the T tubule. This action potential also come in contact with the sarcoplasmic reticulum. This black color structure represents the sarcoplasmic reticulum, and the vesicle of the sarcoplasmic reticulum, that is called cisternae. Cisternae are near the T tubule. All the cisternae of the sarcoplasmic reticulum are always around the T tubule. So when the action potential goes through the T tubule, the cisternae get the action potential from the T tubule, and the cisternae are activated. When the cisternae are activated, what happens? The cisternae are filled with calcium, and it liberate all the calcium to the surrounding. It secrete all the calcium to the surrounding. With the cisternae of the sarcoplasmic reticulum secrete all the calcium to the surrounding, the concentration of calcium inside the cell is increased, and when the concentration of calcium inside the myocyte is increased, what happens? This calcium goes to the myofilament, or to the myofibril. When this calcium go to the myofibril, what happens? As I have told you that in the myofilament or the myofibril, the actin consists of three types of protein. This is the actin. This consists of three types of protein. One protein is the black in color, that is actin, or F-actin. F-actin is black in color, and that is bead shape. The second protein, that is troponin. Troponin is having three domains: the domain for calcium binding, and domain for the tropomyosin binding, and the domain for actin binding. So this tropomyosin is also represented over here in the form of thread. This green color represents the tropomyosin. So the the F-actin contains some active sites. These are the active site. This is the active site. This is the active site. This is the active site, and this is the active site of what? These are the active site of actin filament. So when this F-actin, or when this actin is in normal position, this is in resting position, and when calcium is not connected, or when calcium is not combined with this troponin C, what happen when calcium is not combined? These active sites are covered by the tropomyosin. The tropomyosin always cover the active site. You can see over here that the tropomyosin covers the active site of this total actin filament. But when the calcium combines with this troponin C, what happened? The troponin C is activated, and when the troponin C is activated, the conformational changes occur inside this troponin C. And when the conformational changes occur inside this troponin C, the whole troponin goes through conformational changes. And when the conformational changes occur inside the whole troponin, what happens? This conformational changes is also transferred to the tropomyosin. The tropomyosin also go through conformational changes. So the tropomyosin, when go through conformational changes after activation of troponin, what happen? The active sites which have been covered previously become uncovered now, become open for binding, become uncovered. In the previous, you can see that the tropomyosin have covered the active site of the actin F, or the F-actin. The F-actin are having active site, and these active sites have been covered in the previous. In this diagram, you can see that this has been covered by tropomyosin. But when this troponin is activated by the calcium, these active sites become uncovered. These active sites become open. So what happened when these active sites become uncovered? These active sites are now open for the myosin filament to get connected with these active sites. This is the actin filament, and this actin filament are having large number of active sites, and these active sites are now uncovered. This is the myosin filament. This is the tail of the myosin filament, and this is the head of the myosin filament, while this is the arm of the myosin filament. This is hing 1 of the myosin filament, and this is the hing 2 of the myosin filament. The hing 1 of the myosin filament help in the formation of cross bridge, or the hing 1 of the myosin filament help in the combination of this head with the active site of the actin. The hing 2 of the myosin filament help in the formation of power stroke, or help in the moment of this myosin filament over the actin filament. So this head of the myosin filament consists of the actin binding site and the ATP binding site. Over here you can see that this is the actin binding site, and this is the ATP binding site. So when the active site on the actin filament become open, what happens? Or become uncovered, what happen? This actin binding site combine with this active site, and when this combine with the active site, what happen? You can see that it make a bond. This combine with the active site, but what happen? This hing help in the formation of strong link through this hing. This hing gave a good moment to this hit to make a strong cross bridge. So this hing is implied over here for the formation of cross bridge. So what happened when the bond, or when this head is combined with the active site on the actin filament? Once again, when this head of the myosin filament combine with the actin active site, what happens? The ATP combine with the ATP binding site over here, and the ATP binding site contains the ATPase enzyme, and that ATPase enzyme break down the ATP into ADP. And when the ATP is broken down from the ATP to ADP, the ADP is still attached with the ATP binding site, and the energy liberated is transferred to the head of the myosin filament, and that energy is utilized by the head of the myosin filament to twist around this hinge, and the twisting of the head of the myosin filament around this hinge help in the sliding of the myosin filament against the actin. You can see over here that the angle the angle is completely changed. The angle is completely changed. The angle, this angle, the angle of the tail and the angle of the arm. And over here you can see that the angle of the arm and the angle of the tail, and over here the angle of the arm and the angle of the tail, that is completely changed. This stroke is called the power stroke. This energy is provided by the ATP, and when it gets a power stroke and when it slide against this ATP, when this slide against this actin, what happens? The ADP is also detached from the head. When the ADP is also detached or separated from the head, what happens? This head is also separated, or this head is also detached from this binding site. So after the pause stroke, what happened? After the pause stroke, the ADP is also separated from the ATP binding site. When the ADP is separated from the ATP binding site, what happens? This head is also detached from the attachment site. When this head of the myosin is detached from the binding site, what happens? This goes to its original position, and when it goes to its original position, what happens? Once again, this head come in contact with another active site, as the actin fiber contain large number of uncovered active sites. So it once again it come in contact with another active site and make a strong bond, and the bond formation or the attachment of this head strongly is supported by this hinge. So this hinge support the strong bond of this head with the actin filament. So when the strong bond is formed between the myosin head and the actin filament, what happens? Once again, the ATP combined with the ATP binding site, and when the ATP combined with the ATP binding site, once again this ATP is split into ADP and inorganic phosphate, and the energy is liberated into the head. And after the liberation of the energy into the head, this energy is utilized by the head by twisting around this hinge, and this twisting around this hinge help the sliding of the myosin filament against the actin filament, and this process continues throughout the cycle. This process continues, as you can see in this figure. This is just like this one. So this process continues.